Hi, I'm Jake. I'm presenting my poster titled Widespread Redundancy in Omics Profiles of Cancer Mutation States. So the general research questions we wanted to answer are sort of comparing omics data types for capturing functional signatures of cancer mutations. In particular, is this dependent on the sort of gene or mutation that we're considering? Or is there sort of a general trend that one omics type is better than the other that holds across a bunch of different cancer genes? And we also want to know if we can combine multiple omics types to do better than single omics types. So can we build a better classifier or a better predictor of mutation state by combining, say, expression and methylation? So we can formalize this as a prediction problem where the labels are the presence or absence of a genetic alteration in a particular sample. And the features come from functional or omics readouts. So in particular, these are gene expression, DNA methylation, protein abundance, microRNA, and mutational signatures. And we get these from TCGA. We're going to use processed level 3 data, and we'll use pan-cancer data, so across all of the different cancer types in TCGA. We can also use TCGA to generate the labels. We can use combined non-silent point mutations and CNV info to generate a binary label for each sample, whether it is mutated or not mutated in a particular gene. To evaluate performance, we took a large set of cancer genes to generate our labels. So we took 280 different cancer-related genes from 3 large survey studies. Most of these are sort of pan-cancer drivers or really known to be relevant across multiple cancer types. And to evaluate performance, we're going to do stratified cross-validation. So we split our data into training test sets that have equal proportions of each cancer type in TCGA. And we'll do that 8 times, so 4 folds times 2 random seeds. As a performance metric, we used AUPR. There are 2 reasons for this. It handles continuous output well, which comes out of our elastic net classifiers that we're using. And also works well for imbalanced labels. So there are a lot of genes where there are only a few samples that are mutated and many that are not. And AUPR can be used to make meaningful comparisons in these cases. As a baseline, we want to look at a model with permuted or shuffled mutation labels. The idea behind that is it controls for the predictive effect of cancer type on its own. And we want to sort of look at predictive ability that comes from the omics data above that sort of baseline. So that's what we're going to look at. So this plot shows performance for expression and methylation on their own. We take all of the samples in TCGA with expression, 27K methylation, and 450K methylation. That gives us about 8,000 samples. The columns here are those 3 data types. And on the y-axis here, we see performance above the shuffle baseline. So higher is better. Each point is a gene. And we can sort of see that the performance for gene expression tends to be slightly better. So the mean performance is a little bit higher for gene expression, suggesting that across all of the 280 genes, it does do slightly better. When we include all of the omics data types, we have less samples now because every sample has to have every data type. This gives us about 5,000 samples now. And again, higher is better. So we can see that gene expression continues to do slightly better than the other 5 data types in this case. So it tends to be sort of the best default choice across all of the genes. So next we ask this question about multi-omics. To train multi-omics classifiers, we're just sort of combining data types into one large feature matrix, just sort of concatenating them, and then training our elastic net models on those concatenated feature matrices. So in this case, the teal boxes are the best single omics predictor for that gene, where the columns are genes, and the orange boxes are the best multi-omics predictor for that gene, from combinations of gene expression and the 2 methylation data types. And so to compare these statistically, we can do t-tests. I'm just sort of comparing the single omics and multi-omics models over the 8 cross-validation folds. And we see that none of the differences between single and multi-omics are significant at a cutoff of 0.05. So for these genes, we don't see that we can train better multi-omics classifiers statistically than the best single omics equivalent. So in general, what have we learned? We saw that gene expression tends to capture the most signal overall across the 280 genes. But we see that mutations that have strong functional signatures do tend to perturb many or all of the omes. So if you look at a strong sort of pan-cancer driver, something like BRAF or P53, we can see that most of the data types or all of the data types can be used to train a good classifier. So the sort of signal from those mutations gets propagated through almost all of the data types. And that was sort of interesting to us. And we also sort of surprisingly saw that there was no discernible difference between the best single omics and multi-omics models for these 6 genes that we picked. So statistically, the best single omics predictor seems to do as well as the best multi-omics predictor in all of these cases. So we have a paper that recently came out in Genome Biology. The link is here. All of our code and analyses are on GitHub here. And my contact information is on my website if you'd like to ask questions or chat about this work. Thanks.